So, good afternoon and Happy New Year to all. This is the January session of the young seminars organized by the Italian Society for Statistical Physics, and today we'll have just one speaker because the second speaker had some major problems that prevented him to be here, unfortunately. So today we have a talk given by Marta Biondo from the University of Catania and the University of Palermo, and just a few words to present her. In 2018 she got a master in physics of complex systems at the University of Turin, working with Matteo Zella on first passage time problems in gene expressions, the role of extrinsic noise. Then in 2023, she got a PhD at the same university, working with Osella and Michele Caselle, working on exploring the multiple layers of cellular variability. Then she had a one-year postdoc in Turing, and since March 24, she's now a postdoc at the University of Catania in Palermo, and today she's going to give a talk with the title "The Intrinsic Dimension of Gene Expression During Cell Differentiation." So, please, Marta.
I start sharing the slide. Thank you very much, Professor, for your kind introduction. And I also want to thank the other organizers for giving me the chance to share my work today. As you already said, now I'm in Sicily working on transcriptional data for the study of plasma cell cancer. But today, I'm going to talk about this work I did when I was at the University of Turin, and I worked with these wonderful people that are here in the picture. And this work was how I started dealing with transcriptomic data in the first place. If you're interested in this kind of study, the preprint for this work is already available on my archive, and that's the QR code. Okay, then as suggested, given the high interdisciplinarity of these seminars, this series of seminars, I'm gonna use the first part of my talk to give you a very basic biological introduction on gene expression and on what is cell differentiation and why is statistical physics important when studying this phenomenon. Okay, in this picture, there are three cells. Okay, cells, as we know, are the building blocks of life. And even if cell theory is now like 300 years old, in the last, let's say, 20 years, there's been kind of a revolution in this field given by an unprecedented technological advancement, and now we can look at individual cells with an extraordinary resolution, and we can characterize cell heterogeneity, not only with microscopy as in this picture, but especially with transcriptomic, proteomic, and even multiomic modalities. However, if we forget about this impressive amount of data that we can get from single cells, and we wanna draw a minimal model of a cell, we can get back to the central dogma of biology, which is gene expression. Otherwise, the process by which a cell uses the information encoded in the DNA to create proteins, which are functional products of gene expression. And it's a two-stage process involving transcription, which takes place in the nucleus, and translation. And of course, it's not that simple because not all the genes are readily, let's say, available all the time simultaneously, but you have to have an accessible region of the DNA, which is open chromatin, as we say in biology. But in the, let's say, minimal representation of a cell, we can think of it as a bag containing basically RNA strains and proteins. And in complex organisms, every cell has the same DNA, and so each cell can be considered a unique realization of the same DNA. What gives the cell special function and properties is the fact that they express a certain set of genes. And specific cell types, we have hundreds of them in our body, express specific set of genes selectively. But however, as I said before, the DNA is the same for all of them. So it should not be so much surprising that all the different cell types and all the phenotypic heterogeneity that we have in complex organisms derive from one single cell type, the totipotent stem cells. And cell differentiation is basically the process by which via cell duplication and differentiation we got from stem cells to fully functional committed cell types. And it's apparent that these happen hierarchically, especially during the embryogenesis. We start from one fertilized eggs and we get billions of cells specialized, and this is a progression by stage and by bifurcation and not necessarily dichotomical choices. And cell differentiation, however, also take place when the organism is fully formed. And also adult organisms have a compartment of stem cells for tissue homeostasis maintenance and tissue turnover in case of an injury, for example, for epithermal homeostasis. However, now this field is really interesting to me, also because it has been proven that cell differentiation is inducible and also reversible, at least in vitro, and this offers an unprecedented, let's say, potentiality in precision medicine. For example, in this paper I'm referring in this picture on the right, they started from fibroblasts, which are fully differentiated epidermal cells, and by using only 4 transcription factors, they are recover pluripotency. And then once you have induced pluripotent cells, you basically virtually can create any cell types, and so you can cure a lot of disease that depends on the failure of a single cell type, for example. However, this is kind of the future of cell differentiation studies, but if we look at the past, let's say, there is this very popular picture of cell differentiation, which is from the late 50s, and it is the Waddington landscape, which basically offer a metaphor for understanding differentiation. This metaphor says that pluripotent stem cells become committed to a specific fate gradually, and let's say spontaneously, just like marble rolling down a hill, following this differentiation potential, and finally settling down into attractor basins, which are the differentiated cell types. So with this picture in mind, it's clear that the statistical mechanics have a lot to give, both in terms of modeling and interpretation of data, because looking at this picture, we all think about any kind of state manifold or energy landscape. And in particular, it's clear to us that the cells are like microstates, and tissues or organs or organism itself are macrostates, and so the analogy is well set, let's say. But what are the configuration space for these instances for cells? Well, this is the most famous picture from Waddington, but in the same figure, the second panel was this one, where he proved to be, let's say, ahead of his time, because he proposes that what shapes this energy landscape is indeed gene expression. So there are these pegs that are genes, and there's also the notion of regulation and genes interactions with these network representation. Now, nowadays, there are protocols that allow us to access the gene expression at single-cell resolution. I'm talking, for example, of single-cell RNA sequencing protocols, where you start from a tissue or a sample, and you can isolate cells, tag them, and then extract all the RNA contained in them. And RNA, if you remember for the, oh, that's the pointer. From the slides before, RNA is good. It constitutes the phenotypes together with proteins, so it's good to know. But also, it's halfway between DNA and protein, so in a sense, it's a proxy for both chromatin accessibility and the phenotype of cells. So that in the end, when you perform single-cell RNA sequencing, you have the transcriptional profile cell by cells. A fancy way to store this information is using count matrices, where you have genes on the row and cells as column. And each entries represent the number of transcripts of a certain gene in a certain cells. Now, these objects, from the mathematical point of view, are not so easy to analyze, because in the machine learning paradigm, let's say, you have 20,000 features and 100 or thousands of cells, so it's not very straightforward to interpret the results of your experiments. And more importantly, there are many zeros in this object, so they are called sparse metrics, and they suffer from noise and heterogeneity, which has both biological and technical causes, and it's not that easy to distinguish this contribution. Anyway, if we forget about all these challenges and think about count matrices just from a purely geometrical point of view, a cell is a vector, an array, and in particular, it can be thought as a point in a 20,000 dimensional space, which is called the gene expression space, where you have one axis for each gene, and the expression sets the coordinates. Now, we have a lot of data set of this kind, so we can think of clouds of cells in the gene expression space, and this should be the generalized coordinate to which the energy landscape proposed by Waddington refers to. So we want to link this kind of data with this representation of them. And the open questions, which interest me the most in this field, are, is really there is an underlying energy landscape in data. Should I see the signature in these matrices? And in particular, is there any proxy, any quantitative measure that we can extract from data to estimate the differentiation potential of cells without looking at specific marker genes for stemness? In this framework, there are already many models taken from statistical mechanics. And indeed, the concept of stability, for example, I already mentioned it before. And this also relates to classical dynamics when you consider just a few transcriptional factors, but having thousands of them in a complex gene-gene regulation network produces what we call a rugged landscape. And so if we consider spin-glass Hamiltonians to describe this kind of landscape, the principal, and we don't want to go so much in detail, adding, you should add a term for each contribution to what can change the cell or tissue status. But the minimal take-home message of this kind of models is that cell types are like basin of attraction, and you can map the transcriptional profile of cells, discretizing them to a spin configuration. let's say gene off, it's spin -1, as gene on, it's the active state. And also there is this mapping between temperature and potency, because you can think that stem cells, which prioritize plasticity, accept changes of the status, the configuration, without losing their functions, while if you cool down your system with differentiation, you slowly fall in this basin of attraction, which are differentiated cell types. So our hypothesis, the intuition we're gonna test, having this in mind, is that if temperature sets the potency level, and you can think of differentiation as an increase of geometric constraint and correlation, you do expect with differentiation a progressive reduction of the intrinsic dimension. In particular, this is a cartoon where we have a data set in a three-dimensional gene expression space. If you can express each gene at any level, you occupy it uniformly, but if there are biological restriction and gene regulation, you have correlation among genes, and so your data point should lie on a manifold with the reduced dimensionality. So what's changing here is the intrinsic dimension, and which is 3 on the left and 1 on the right, and there are many estimators to evaluate the intrinsic dimension of a given data set. The most simple of it is based on principal component analysis with a diagonalization of the gene covariance matrix. You can look at the number of principal components that explain a given threshold of variance, and this is an estimation of how your data points are spread and what's the dimensionality of the bending they came from. But also you can look at the entropy of the eigenvalue distribution, and this relates to intrinsic dimensionality as well. The estimator we used in our work is called two nearest neighbors, and it basically looked at the statistic of the ratio between the distance of each point from the two closest neighbor, as the name of the algorithm says. And it's super fast, and it's well suited for single cell RNA sequencing data. So the results I'm going to show you all leverages on this estimator. Before going on real data, we decided to test our intuition about the decrease in intrinsic dimensionality in a toy model of a Waddington landscape. And this toy model involved an off-feed neural network as a spin glass system. And we tried to match the features of this network with the typical order of magnitude of things in single cell RNA sequencing data. For example, genes that represent the spins in our simulation were thousands. And basically, when you perform single cell analysis, you may consider 1,000 highly variable genes. So it's a good match. And we are generating a landscape involving 10 cell types, which in the case of off-field model are the minima or the attractors. And our check was right, say, because decreasing the temperature, which is going in the direction of differentiation, you observe a decrease in the intrinsic dimensionality. And so once we tested our hypothesis in this toy model, it's time for us to look at real data. And there are many data sets available online in which they've performed sequencing at different stages of embryonic development. So you have one matrix for each embryonic day of mouse gastrulation, for example. And what we observe, once again, is that here on the plot, on the x-axis you have time, on the y-axis you have the intrinsic dimension as we estimate with 2nn. And you observe that there is a decreasing trend, as we expected. And this disregards the 2nn algorithm, but also using PCA-based approach, we observe consistently a decrease in the dimensionality. And we can also evaluate correlations, constructing this network of gene-gene correlation, and the number of genes involved in the network is increasing in time. So everything seems to match our first hypothesis, and the fact that the intrinsic dimensionality reflects the differentiation level of mixed cell population. And now in this red plot, there's just scaling, because we are not really interested in the fact that the intrinsic dimensionality is 70 or 50, also because this number depends on the number of cells you use to estimate it. So we would rather scale this quantity to produce this ID score, which is intrinsic dimensionality score. This was for a data set of mouse gastrulation, but we verified it in another dataset about different model organisms. On the left, we have the zebrafish, and the right, C. elegans. And even in those cases, you can observe this decreasing trend. And it is independent also of the time frame the time window you're exploring. The plot at the bottom refers also to zebrafish, and in different time frames with respect to the one on the top, and also regarding the data set of represented in light blue, it's about mouse postnatal development. So even after embryogenesis, during the postnatal development, you can still recover this decreasing trend, and seem quite robust, really. Sorry for my throat. Okay, so there is this invariance independently of the time window you're exploring, but also an invariance in the scale of the tissue you're looking at, because previous plot refers to the entire organism. But if we focus on a single organ at the time during the formation, we have again the decreasing trend during the formation of the pancreas, of the neural cortex in mouse, on the left in the middle, and in zebrafish during the formation of the neural system. So once again, the ID reflects the differentiation of the organs. And also in postnatal development, each color on the plot at the bottom represents a single organ. And if you compare all the couples, there is a decrease between the fetal and the oldest stages in mouse in terms of intrinsic dimensionality score. Okay, so everything up until now seems to work. Indeed, seems to confirm our hypothesis, but we must not forget that during gastrulation and organogenesis, we assist to proliferation in the number of the cell types. Sorry. Okay, just look at this plot, should not be there. This is the cell type composition for mouse in the time frame we analyzed. And we see that at the beginning, we only have the epibus, the mesoderm, and the 3 germ layers, let's say, but then the number of colors increased dramatically. And this is for mice, but this plot here refers to all the other developmental data set where there is a correlation between the intrinsic dimension and the number of cell types. And this is independent of the level of differentiation of single cell types. Indeed, to test this, we tried to create these synthetic samples mixing defined cell types, and we observe this decreasing trend. This is intuitive when we think about intrinsic dimensionality as in the picture I showed you before, because if you only have one, let's say, cloud of points spread, you have 2 important directions. But if you add another cell type, and we remind that cell types are clusters in the gene expression space, adding another cluster far away from them means that the direction that link the two clusters explain all the variance in your data. So if you estimate the number of principal components with 2 cell types, you only get 1 instead of 2. It's not easy to evaluate if the intrinsic dimensionality is decreasing because it reflects the overall level of differentiation of cell types, or maybe it decreases just by the fact that we are multiplying the number of cell types. And instead of evaluating these two contributions to the decrease of the ID score, we want to try to rule out the having multiple cell types in a single matrix. And we can do so. We can consider a subset of the count matrices, selecting specific cell types. And there are many datasets available on that, where they also reconstruct the differentiation trajectories. For example, in the dataset about pancreatic endocrinogenesis, they have all these cell types organized in a differentiation tree. At the top, you have multipotent cells. There is a first bifurcation between tip and trunk, and then other bifurcation in the case of trunk cells. And so we measure again the intrinsic dimensionality of matrices containing only one cell type. And what we find out is really interesting, because you can see that the intrinsic dimension reflects the potency of cell types along developmental lineages. In this plot, each color is a separate lineage of differentiation, and there is a decreasing trend, with multipotent having the highest dimensionality score, and endocrine cell, which are the most differentiated, the minimum score, but also the bifurcation are well resumed in this plot. So this suggests a possible application for the ID score after clustering in a single cell analysis. You can measure the dimensionality of your cluster, and this should help the annotation of the cell type, and also should help to build this and to reconstruct these differentiation lineages. Okay, this was one example, but we have many of them, not only in mouse, as the one I showed you before, but also in zebrafish. So in this paper I referred to, they also ordered the cell type by a potency score, looking at marker genes or reconstructing cell lineages. And here, measuring the ID reveals a good agreement with this this effort of reconstructing differentiation trajectories. Basically, the progenitor cells, which are more stem, more pluripotent, are always at the top of this plot with the highest score of dimensionalities, and there is a decrease of cell potency that corresponds to a decrease of dimensionalities in case of fully differentiated cell types, such as erythroids or excitatory neurons, and as well as in the other two examples. Both these figures here refers to cell type differentiation during embryogenesis and development. But as I said to you earlier, indeed, during tissue turnover, there are a compartment of stem cells that are responsible for that. And if you want to study this kind of phenomenon, one model organism is Hydra, which is a pulp that has the peculiarity to have a strong regeneration capacity. And in the three layers that constitute this organism, you can also use the ID score to discriminate between progenitors and differentiated cell types. And it's a very clear distinction between these kind of cell types. So with that, I conclude. This is the last slide. And I listed here the take-home messages. I don't know if I've done, yeah, different means. So basically, I hope I show you how tools and ideas for statistical physics are really meaningful to address single cell data and to interpret them. And I show you that it's fruitful to explore the geometry of the gene expression space, and in particular, the dimensionality analysis support this picture of the Waddington landscape. And also that the intrinsic dimension reflect the cell potency at cluster level, not cell by cell, but considering an entire data set, and a score that is robust, quantitative, and more importantly, does not rely on prior biological knowledge, as it's the case with the standard pipeline of analysis of single cell data. And from a more practical standpoint, I show you also that the two nearest neighbor algorithm is very, very valid to perform this kind of analysis, and that it should be a good step to use this kind of estimates to implement intrinsic dimension estimation in the standard workflow analysis of single cell data. So with that, I conclude. Thank you all for the attention. And once again, I thank my tutor, my prof, and my co-workers in Turin. And that's the link to the preprint.
Okay, thank you, Marta, and thank you for your talk. So, is there any question or comment?
Yeah, also I should mention this work is still going. I'm still working on this, so if you also have just, I don't know, feedback or suggestions on that, it will be really more than welcome.
Actually, I have a question. I mean, when you started to present your statistical mechanical modeling, you made a parallel between temperature and potency. And you have also shown a figure plotting the intrinsic dimension as a function of the temperature, showing an arrow towards lowering the temperature. It is the differentiation process. But I wonder, is there any way to think about a sort of reverse process? So when the potency actually increases.
Yeah, it's a super interesting point. And actually, we explored this this scenario because you see when you decrease the temperature and you have a fixed number of minima, it's like in the real data when you have multiple cell types. So you don't know that the fact that you are reducing the temperature and measuring decreased dimensionality is given by the fact that you have multiple minima. But actually, we also perform this different set of simulation. When we start from a low temperature, we increase the temperature to see, starting with a single minimum, what we got is the increase of dimensionality. So perfectly right. Actually, I'm showing you, I'm still showing the slides, right? So this other plot where you have a single minimum and you recover the intrinsic dimension. Can you see that? The plot on the right.
Okay.
This was produced increasing the temperature.
Okay, but so you run the same. I mean, so you have the same plot, both decreasing and increasing the temperature.
Yeah, I mean, no, the same result. What changes if you have one? Yeah, we can increase the temperature and escape the minimum, the single minimum.
the difference between the two graphs that you are showing?
The set of simulation are similar, and the temperature is tunable. So it's you generate a data set for each temperature you set. So it's really not that important if you're going from low temperature to high temperature. Point is that the temperature sets the arrow of differentiation, but it's just for interpretation. From the point of view of just running the simulation, you fix the temperature, and we have multiple points for every value of temperature which we choose.
Okay.
And the fact of increasing the temperature is important to escape the minimum.
Can I make a question?
Yeah, sure.
Hello, nice to meet you. Thank you very much for the talk.
Thank you.
Like a very naive question. So the fact that this intrinsic dimensionality decreases as differentiation happens, does it mean in a certain sense that like less genes are becoming important for the differentiated cell compared to the multipotent cell? So like in a certain sense, these plots that you show, they are more stretched, and there are less directions in which like differentiation is important.
Yeah, maybe the link with the purely, let's say, the number of involved genes, not the straightforward, but because yeah, there's not a way to you can also measure the number of active genes, let's say genes that are on.
Exactly.
There is actually a paper in which they clearly states that the number of active genes should reflect potency. Maybe I have a slide on this. So that's an interesting point. Yeah, this is the paper. They propose this cytotrace, which simply counts the active genes. This gene counts refers to genes that are not zeros.
Okay.
And in certain case, there is this very simple trend that reflects differentiation. But in our experience with the dataset we tested, this measure is not robust because yeah, I mentioned it, but maybe I was not clear enough. I didn't spend so much time on that. The fact that these data are really noisy, it's also given by the fact that when you capture RNA, you know, it's not the same for all the cells. You have what is called a sequencing depth, which is how much you can sample for a cells, and this cell size, the number of time you sample for a cells varies a lot. So the number of zeros that you get in the transcriptional profile really depends on the quality of your sampling. It's not robust. For example, here in mouse gastrulation, in time you don't have a trend of the number of detected genes, so the number of genes that are on. And this trend is even worse if you only consider cells with a given number of reads per cell, so trying to minimize the variability in the sequencing depth. If you consider only a subset of cells, this is even worse. It's just noise. There's no signal. This is about mouse gastrulation, but we also tested it in lineage, in differentiation trajectories. So not for the entire embryo, but for specific lineages, and it doesn't work really well.
I see. Oh, because
but it's a good point. I mean,
I still miss a little bit of intuition on why these intrinsic dimensionality decrease towards differentiation should like confirm the Waddington landscape, like
Waddington landscape.
Like
yeah,
because
if you
on the like in the second part of the talk where you compare like differentiated cells, not like all differentiated cells in the tissue.
Yes, so you want to
no, like because exactly
sorry,
like
I missed the audio for a bit. just to cover your questions, it's about the interpretation of the decrease in dimensionality. Yeah, if you, I don't know if you're familiar with this rough energy landscape, but a cell is an entity. I think so. Otherwise, and temperatures in our simulation, for example, temperature together with the decrease of energy is how much you are prone to accept a change in your transcriptional state, so in your configuration of spin. And if you are plastic and your only function is self replication, you allow any change in your transcriptional state. You express many genes, but at low levels, so you don't see the landscape because you navigate it without seeing the minimum. The temperatures could dip down and force you to get to the minimum, and cell types that are differentiated, they don't allow for plasticity, because if you cannot perform your function, the organism abolish you. So it's there is a narrowing of the accessible state in the configuration space. I don't know if I'm...
Okay, I get it. Thank you very much.
Thank you.
Okay, thank you.
Maybe there is Irene with...
Yes, sorry, can you hear me?
Yeah.
Yes.
Okay, so I have a very quick question. Did you test human data, for example, stem cells of some kind?
Not yet, no. But
okay.
We plan to do this next.
Okay.
Sorry.
Okay. Is there
just the question was about human data? Are you in the field of? May I ask where the question came from? Because we are looking for data
from a very different perspective to stem cells. So I was curious whether there are analyses of the investigations of this kind on stem cells, pluripotent cells, and differentiation in human cells.
We just started analyzing this data set, which should be an atlas of human embryogenesis, but as you can imagine, there are, let's say, ethical issues with that. So there are not so many data sets. The one we are studying has been produced in Munich at the Helmut Center. There is a paper on that, but still a work in progress for us.
Okay, thank you.
Thank you.
Okay, so if there are no other questions or comments, I thank again Marta and also all other people interested in her seminar. So thank you again to be here, and so let's see again in February as usual, the second Thursday of the month. Ok, bye. Ciao, saluti a tutti.
Bye, buona serata.
Ciao.